Hello, everybody. We're really, really excited to get away, so let's get underway.
All right, so it's truly a dirty trick of the organizers to not only make you listen to me twice, but do it back to back. Okay, so you've heard a little bit about so-called stem cell-based embryo models. And what I'm going to talk about today are the sort of broader problems of approaches to governance of them, in a way that's actually consonant with what I talked about in the last panel. And I want to begin by pointing you to two pieces of black letter law. This is Article 18.2 of the Oviedo Convention that I was just talking about a few minutes ago. Okay, and here is Dicky Wicker. This is U.S. law governing federal funding for, prohibiting federal funding for research involving harm to human embryos. But I want to in particular draw attention to the definition. Human embryo or embryos includes any organism not protected as a human subject, et cetera, regardless of where it comes from. So human diploid cells is the sort of key biological modifier of any organism. So, in a moment in which embryos sort of could only come into being through the fertilization of egg by sperm, this is a pretty straightforward definition. It gets a little bit more complicated with cloning and parthenogenesis and so on and so forth. But it's now basically immensely complicated because there's been an emergence of a repertoire of techniques that have basically departed from the project of stem cell research circa the 2000s, which was a kind of mechanical directing of cells towards cell fates, which turned out to be, pretty much not possible. A reorientation towards using the niche, using the surrounding organismic environment as the supporting structure that guides whatever your desired cell or tissue is towards its fate. In other words, the organism is the best bioreactor for producing its parts. And that's the kind of research orientation, which it seems to me is a lot more like gardening than engineering, right? We don't actually understand or control the vital potencies in the biological entities that one plants in one's garden, and yet if one manages their environment and provides them with the right sorts of inputs, you can get them to grow. So the organism in that language does an enormous amount of work. And the distinction that matters here is an is not distinction. It is or it isn't. And yet the enormous flexibility about what can be produced, because this is not a category of fixed things, rather it's a repertoire for creating, a sort of roughly infinite or vastly expansive set of possible human biological configurations. The is not distinction becomes the crucial distinction. In effect, this becomes the question, what is it? What is it? The ethical question becomes transmuted into the question, what is it? And what is it is, you could say it's an ontological question. In practice, it's a technical scientific question. It's a question for expertise. And so this is what I want to focus on here, the problem of how a regime of governance, which is in effect what we have currently, at least in this country, with spillover into a variety of others, what it means to have a regime of governance that is organized around definition. And I want to make 3 points. Number one, that ethical discernment becomes technical evaluation of is not, which is to say there's a shift from a conversation which is normative or normatively inflected and therefore intrinsically inclusive or should be inclusive, to a conversation which is over which those who have the most refined knowledge have jurisdiction. That is to say, the people who are in the business of making these things. Number two, that in the move to that kind of technical evaluation, where the notion is that that technical evaluation is the locus of fundamental sense-making that informs ethical evaluation, there comes to be a kind of elevation of science, of scientific expertise, of scientific knowledge, as arbiter of public reason, of the terms of debate. So in effect, what's getting evaluated is not just the is notness of a biological construction, but the is notness of informed public ethical deliberation. And that the consequence of this is constrained moral imagination. That's basically the argument that I want to make. So it's not that the definitional questions aren't important, it's that the definitional questions are delimited. okay? Okay, just a little illustration here. Oops, shoot, my movie's not working. This is supposed to be a gastroloid that is exhibiting forms of spontaneous self-organization, but sorry about that. This was from a conversation that I had with a researcher who had made an early gastroloid about a little less than 10 years ago, who captures the sort of problem of governance. I ask him, is NIH worried about these things? He says, I got to have these monthly calls with the NIH policy people and the lawyers, and they ask me, is this an embryo? And I say, what do you say? And he says, well, I tell him, no, there's no evidence these things are embryos because there's no evidence they can develop. But you know why? Because I haven't done the experiment. And he kind of cackles. So here is an image, here is a video, a movie of one of these things. And here are a series of accounts. You could pluck any number of these from the public discourse that are not only characterizing it as not an embryo, but characterizing it as irresponsible to suggest that it is anything but not an embryo, okay? And yet, at the same time, there's a kind of paradox here, because the notion of the model is that the model is as close to the thing as you can possibly get. So here are two very competitive people, both of whom I'm friends with, incidentally, where one is saying, the other one's is crap, because it's not really a good model, because it's not very close to the embryo. You really want it to be basically really close to the embryo, right? But there's a sense in which there's been a kind of definitive declaration of the nature of the things, informing, as a sort of line in the sand, informing public deliberation. And that's gotten codified in what is effectively a set of guidelines that function as the law of the land, for instance, at this university and many others, and the international journals and so forth, about what distinguishes ethical from unethical research. I want to point out how there's been a kind of expanding jurisdiction that has increased discretionary judgment within spaces of oversight, behind closed doors oversight, that rely upon scientific expertise. So basically, there's been a movement to drop limits, and generally speaking, a move towards evaluating things through review. It's ethical if it's been subjected to review. The only limits now, the categorical limits on the things you can't do with stem cell-based embryo models, are transfer them to a human or animal uterus, or gestate them ex vivo beyond viability. So that's pretty permissive, right? A corollary to this sort of question, what are they, is what should they be called? And this sort of speaks to the ways in which public reasoning is reached into. So here's from the International Society for Stem Cell Research that advises against using the term synthetic embryo because it's inaccurate and can create confusion. Their integrated embryo models are neither synthetic nor embryos. We've seen this movie before, so wind back the clock about 20 years, and the ISSCR intervened in discussions around cloning by trying to change the terminology that was used in order they replaced therapeutic cloning with somatic cell nuclear transfer for the procurement of stem cells to introduce scientifically accurate terminology to facilitate frank, scientific, ethical, and public debate, et cetera. Suffice it to say, I'm going to skip a couple of these, but these interventions were made not just in the name of scientific accuracy, but in the name of the quality of public deliberation. So it was like an intervention into democracy in order to safeguard it from itself, right? Where the claim to jurisdiction, the authority to intervene, is based on the notion that we start with correct definitions, and if we don't start with correct definitions and the corollary forms of terminology, everything else goes to hell. This is actually where my heart is, so I want to spend my last minute on this. The stakes here are much bigger, I think, than getting the definition right, as if all then follows from the definition. Because it seems to me that what is being developed is not, again, merely a set of static things that one has to classify and taxonomize, but a way of relating to human being, a way of relating to human biology, which is in effect a way of relating to human being. And the territory of humanness that is at stake here is very basically biological. It's procreation, et cetera, but it's also deeply significant. And the uncanniness of this headline from Antonio Regalado, who you guys now know and love, kind of captures this, I think. This is a factually correct statement that also kind of elicits a sense of the uncanniness, the strangeness of what's being done in this domain. Here is Jacob Hanna, who I respect enormously and is a good friend, trying to think through how he can do the kind of work that he wants to do without being transgressive. And he's thinking about how, well, maybe the way to do that is to engineer my human-like embryos to be headless, to be truncated in some way such that they fall short of the fullness of the category of humanness. Defining limits in a kind of technical way to allow experimental creativity also is a kind of delimitation, a definition of humanness. I see that I'm over time. So let me just wrap up with this kind of key point, that I think that thinking, the forms of thinking that are happening in the frame of definition are producing a sort of repertoire of how we can imagine less than human, such that we narrow down and settle in upon sort of constructions of human quintessence that allow us the most significant degrees of freedom in terms of the human biologically but not human morally constructs that we can make. And we should be concerned about the ways in which we are engineering basically our shared moral vocabulary in doing that.
Great, big round of applause. Now we'll hear from Vice Provost Condic.
All right, so I'm going to be talking to you about something we've heard a lot about, synthetic embryos and in vitro gametogenesis. The field of assisted reproductive biology, or ART, has advanced significantly since the first test tube baby was conceived over 40 years ago. Yet, despite the proliferation of new reproductive technologies, the overall success of ART has remained relatively constant for over a decade. Moreover, current ART methods do not address all of the situations in which individuals are unable to have a child. Consequently, there is ongoing research into technologies that can circumvent an ever wider range of the causes of infertility, with some of the most intensive research being the research we've heard a lot about today, research into the development of artificial gametes and synthetic embryos. And I'd like to spend just a few minutes talking about the state of the science in both of those areas. So scientists have investigated the potential of isolated cells to reproduce various aspects of normal embryonic development since the early 1900s, when it was first demonstrated that animal embryos could be completely dissociated, and then the cells recombined to reconstitute a living embryo that proceeded normally through development. In modern times, scientists typically culture stem cells under conditions that promote the formation of specific embryonic cell types, and then combine these cells into clusters, known as organoids, that are able to generate a relatively normal but limited set of embryonic structures. So groundbreaking work in 2022 demonstrated that a far more comprehensive level of embryonic integration was possible for such cultures. Research by independent groups showed that when mouse stem cells were induced to assume states similar to very early embryonic cells, in rare cases, they self-organize to generate an entity that undergoes many of the natural processes of embryonic development, including the formation of the primary embryonic axes, the generation of well-organized brain and muscle tissue. Similar but far less convincing results, in my mind, have been obtained using human stem cells. In a related line of research scientists have worked for decades to produce gametes in vitro. Yet, due to some fundamental technical constraints, these procedures could not be readily translated to humans. However, recent work has radically changed this landscape. In 2023, a research team working entirely in vitro, in rare cases, were able to produce functional oocytes from male mouse stem cells. In even rarer cases, these male egg cells were able to produce embryos, with approximately 1% of the transferred embryos maturing into fertile adults. So the ability to produce functional eggs from genetically male stem cells really makes this procedure a game changer. If it can be adapted to human cells, this approach would allow the production of human infants using stem cells obtained from infertile couples of any age, from couples of the same gender, from a living person and a deceased partner, from two deceased persons, and even from a single individual. So I'm now going to turn, and I think you're going to hear more about this from our colleague Sonia, but I'm now going to turn to the current state of regulation for both forms of synthetic embryo production and the ethical challenges that they raise. So currently there are very limited guidelines for the use of human synthetic embryos in the UK, and more globally, the International Society for Stem Cell Research has significantly revised its guidelines to include synthetic embryo research. Yet these guidelines are entirely voluntary, and such a poorly defined regulatory and ethical landscape creates troubling context for this research. For example, the UK guidelines note that because synthetic embryos are not considered to be, quote, actual embryos, they're not subject to the regulations that are applied to spare embryos donated for research by parents following ART procedures. Consequently, synthetic embryos could be made in industrial qualities for a wide variety of purposes, including the development, testing, and manufacture of novel chemical compounds by pharmaceutical companies. When considered in this light, the need to arrive at a reasonable judgment regarding the question of what is a human embryo and what rights adhere to it becomes immediately apparent. As the UK Code of Practice indicates, quote, perhaps the most significant point of ethical concern for all parties is the possibility that a particular stem cell-based embryo model may at some point be judged, in fact, to be a human embryo. Yet, what constitutes a human embryo, and how would we distinguish one from an organoid or a stem cell? So several ways of defining an embryo have been proposed, with most of them, in my view, relying on inherently arbitrary characteristics, such as consciousness or viability. A more promising approach that has been independently proposed from biological, legal, and philosophical perspectives asserts that the defining characteristic of an embryo is that it undergoes development to give rise to a mature individual. This kind of a functional definition is consistent with a long intellectual tradition. Aristotle proposed that to know something's nature, we must look at its potentialities, or its powers and capabilities. Potentialities, in turn, are revealed by acts, and the defining act of an embryo is organization, or the inherent capability to autonomously orchestrate development. In Aristotelian terms, embryos demonstrate an active, proximate potency to develop as a human being from the one-cell stage onward. A human embryo does not, quote, develop into a human being at some future point, at birth or at maturation to adulthood, but rather it is a human being immediately, because the ability to autonomously orchestrate maturation is the defining characteristic of a developing human organism. So based on this kind of a definition from inherent capability, are entities produced from stem cells actual embryos? In the case of animal IVG, the answer is clearly yes. IVG-produced mouse embryos meet the gold standard of functional test. They're able to undergo a complete sequence of maturation to generate a fertile adult animal. The case for stem cell-based embryo models depends on in part on what constitutes inherent capability to undergo development. Using current techniques, synthetic embryos do not proceed through the entirety of development, but they do autonomously initiate a normal embryonic sequence and can therefore be considered actual, albeit imperfect, human organisms, that is, human beings at the embryonic stage of the natural human lifespan. Prudence would suggest that if the ethical status of synthetic embryos is uncertain, we should err on the side of entities that may be human beings. And in the face of important values on either side of this equation, this is certainly easier said than done, but the need is urgent. At this point, any restrictions on this research would primarily affect the scientific community without impacting ART, medicine, or the pharmaceutical industry. And consequently, it's important to determine the status of synthetic embryos before these technologies expand beyond their current limited scope. So as a final point, I think it's worth noting that not all possible advances in science and medicine are worth the cost. The expansion of the ART industry over the last decade has been driven in large part by the sincere desire of infertile couples to have a child and the compassionate desire of physicians to provide that child. Many focus on these emotional drivers to argue that there's a need for advances in treatment of infertility, yet ART is also a multi-billion-dollar industry. And there's a difference between a need and a market. Markets are driven by what people want, and people can manifestly want a large number of things they don't actually need, including things that are both harmful to themselves or socially destructive. Providing what people desire cannot be equated with meeting a medical need, and all desires, no matter how noble they might be, should be balanced against the human cost and social cost. Importantly, the cost of ART is not trivial. Multiple concerns have been raised regarding the social justice of the ART industry in light of the very high cost of these procedures relative to the cost of providing basic health care. Increasingly, evidence has demonstrated that ART is unsafe and associated with serious medical problems, both for the mothers, but more importantly for the children conceived through these procedures. And many concerns have been raised about the practice of ART relative to the rights of embryos as human organisms. In considering the development of synthetic and IVG-derived embryos, effort should be made to avoid simplistic and polarizing language of, quote, medical necessity in favor of a more balanced and nuanced discussion of rights, responsibilities, and costs for all of the individuals involved. So I think that I take the cake as being one of the few people who have finished on time and early.
Hear, hear. Thank you. Now I'm going to call on Professor Suter to present a paper for her and Professor Cahn.
Greetings from the Zoom room. Thank you so much. This is fantastic.
Yeah, this really has been an amazing conference, incredible conversations. I wish we had a whole week to do this. But thank you for including Naomi and me, who you know is online. So our talk focuses on the challenges of in vitro gametogenesis, IVG, which I'm not going to describe. Maureen just described it, and it's in our paper. In an environment where abortion is not a federally protected right, and we're moving towards an increasingly pronatalist approach to the world that disfavors non-traditional families. So our piece, whoops, this doesn't seem to want to advance. Am I pushing the wrong button? There we go. Our piece notes that IVG raises a lot of questions that have been asked with respect to ART. So how do we regulate it? Are we going to treat it like IVF? Questions of parentage, particularly with the unique kinds of family formations you can create. Access questions, we're assuming the technology is likely to be expensive in the way that, that IVF is, and how embryo status, which varies across the country as has been discussed, will play out with respect to access embryos that can be created with this technology. However, it also raises a lot of novel questions that are intensified by a number of things, including the ability to create unique genetic relationships. So individuals can reproduce alone if IVG works. Same-sex individuals can have genetically related children, and there can be reproduction with 3 or more individuals, and transgender people can reproduce in ways that align with their gender identity. There's also the ease of, quote, perfecting reproduction by being able to really create a huge number of embryos and do PGT on all of them to amplify genetic selection, the anti-LGBTQ plus movements, and then the constitutional personhood debates. We want to talk about 3 areas that could potentially affect the regulation of IVG, and then talk about the spectrum of potential regulations, and then end by discussing the implications of Dobbs and Skrmetti, which I think have real implications for reproductive justice in this area. And we think that equality, reproductive justice, and family pluralism are likely to be central casualties here. So the 3 areas, there's regulations of ART generally, family law assumptions and family law, and then embryo politics. So as has been already discussed, not a lot of regulation in this area, including established technologies like IVF, which is necessary to reproduce with IVG. There's the Dickey-Wicker Amendment, which means that there can't be federally funded research for IVG, but it can be done privately, as is happening right now. And then there are interesting questions about whether the 2016 Consolidated Appropriations Act prohibits the FDA from approving IVG. So it prohibits things like germline gene editing and mitochondrial replacement therapy because that involves intentional genetic modification. IVG technically isn't that, but would the FDA interpret it in those ways? Family law is also very relevant in this sphere. It has traditionally assumed that there are going to be two parents and or binary gametes. And that genetics, intent, and gestation, while they don't align perfectly, will at least align in some kind of intelligible way. But the technology challenged these assumptions in 3 different areas with respect to solo IVG, same-sex reproduction, and multiplex reproduction. So solo IVG, where a woman could produce an egg and a sperm to have a child that is genetically related to her, one genetic parent, and about 75% shared genes, not cloning. Or a man could do the same thing. That challenges the idea of 2 parents. Same-sex genetic reproduction challenges the idea of non-binary gametes. So yes, 2 intended parents, but they are of the same sex. Note, I'm going to skip that. So finally, IVG would allow for reproduction with 3 or more individuals. 3 is a little complicated. One person would have 50% genetic relationship, but in any other situation, there would be less than 50% genetic connection. Here's an example of 8 people reproducing with 12.5% shared genes, the genetic equivalent of great-grandparents. IVG also magnifies embryo politics, right? The different approaches to what we view an embryo as, and that has implications for possible restrictions on embryo creation, what kind of testing can be done, disposal and destruction of embryos, and research restrictions. And then finally, turning to access, IVG is likely to replicate the same kind of stratified access that we see with IVF in a country where healthcare is not a public right. And that means that IVG would be more likely to be accessed by more favored family forms. So as we consider the potential ranges of regulation that could exist, at one extreme, we could see total bans, like we have with germline gene editing. At the other end, we could have a more laissez-faire market-based approach. But we could also land somewhere in the middle with restrictions of uses and professional regulation. And use restrictions could focus on particular uses, like, say, limit it to medical infertility, but there could also be user-specific restrictions. So there could be restrictions on solo IVG, limiting it only to married people, or limiting it only to married opposite-sex people, or only to 2 parents. And this regulatory landscape that might appear in the future is going to play out against the Supreme Court's doctrine regarding discrimination, which has shifted dramatically in the last 4 years. There's been some hints that it was going in this direction, but with the Dobbs decision in Skrmetti, we see some major notable changes. And what's really key is that both in Dobbs and Skrmetti, the court held that regulation of pregnancy and abortion and regulation of gender-affirming care are regulation of medical procedures. And therefore, they're not viewed as regulations that are grounded in sex-based restriction and therefore lead to rational basis review, allowing for a broad range of regulation here. And so we imagine that if this continues to be the doctrinal landscape, that IVG regulation would likely be viewed as regulation of a medical procedure leading to rational basis review. Now, given that there probably need to be some guardrails with respect to IVG, the question is what kind of regulation is status-based exclusion? And we could imagine regulations that would be aimed at prohibiting solo IVG, IVG with same-sex couples, multiplex reproduction, and transgender reproduction. And how might the justifications, how might the restrictions be justified? Well, you could argue safety concerns. Solo IVG is like consanguinity on steroids and would increase the risk of recessive conditions. Concerns about child welfare might argue for bans of non-marital uses of IVG. And concerns about morality and natural reproduction might be used to justify allowing IVG only for opposite-sex couples. And in the context of perfecting reproduction, it would make it possible, as I said, to really amplify selection by creating a huge number of embryos, which could lead to a number of various kinds of concerns that could lead to regulation there. And so what we think is likely to happen is that there will be a patchwork of federalism, like a patchwork of regulations like we see with abortion right now, where some states ban or criminalize IVG, others restrict or limit it, and then others permit it, subsidize it, or maybe even encourage innovation. So our conclusion is that IVG raises old ART issues at a new scale, that the Post-Dobbs Skrmetti doctrine really heightens the risk of restrictive regulation in ways that affect reproductive justice. The parentage, reproductive justice, and equality concerns really have to be addressed before IVG arrives. We don't want the technology to be driving the law. We'd like the law to come first. And we really want to emphasize that IVG implications go beyond reproduction. They raise questions of health law federalism and reproductive justice. So I tie with you, Maureen, for beating my clock, too.
Woohoo! Next, we're going to hear from Professor Schurz.
Thank you to Glenn, the organizers, for having me at this wonderful conference, to all the other speakers. So the inspiration for this paper really came out of my general confusion. For classical ethics, ethics emerges from ontology. Just as a thing's action follows from the kind of being it is, our duties towards something arise from the need to act appropriately towards it. Thus, determining what a thing is a crucial first step in ethics. My problem is that I keep being approached by people asking me about entities that they're having trouble categorizing. AI has been what I've been looking at mostly, but also things like DishBrain, a computational architecture that combines neurons with computer chips, organoids, and of course, stem cell-based embryo models. This paper is an attempt to address these intersecting lines of confusion and draw some distinctions. Now, we have a rich set of resources for thinking through whether an embryo is an organism, and thus has all the kinds of rights that a human organism should have, and these are developed by people like Maureen Condic. But all of that work considers human development unfolding from a single cell emerging either from conception or something like somatic cell nuclear transplant. And this is what Emma Waters was talking about earlier. In contrast, SEBEMs are assembled from groups of cells. By combining stem cells in 3D culture, researchers are generating models, as we've heard, that are like blastoids and gastruloids that recapitulate aspects of early embryonic organization. So a second inspiration for this paper was a debate surrounding the production of COVID-19 vaccine from cell lines that were derived from tissue from an abortion. One set of arguments pointed to their artifactual nature as meaning their source was ethically unimportant, and this was a kind of line developed by Melissa Moschella. While I agreed with these arguments around the liceity of vaccines, calling these cells artifacts didn't seem quite right. However, given my work on AI, the category of artifact seemed promising. So what I started to do and did in this paper was to investigate whether this category of artifact truly applied to these kinds of various entities we're discussing here. So the key distinction for my paper is between substance and artifact. For Aristotelians, substances are the basic units of being, the basic furniture of the world. They're unitary things that, though enduring change, retain their identity. All Aristotelian commentators agree that living organisms are exemplary substances. Though organisms constantly change, grow, develop, eat, and excrete, they retain recognizable identity over time. The substantial form, what in animals Aristotle calls the soul, provides the principle of unity through change. This form is the internal organization of the organism, meaning that the organism is self-organizing, having within itself a principle of change and development, rather than merely being acted upon by others. And this is what, in part, gives us its unity. Further, the organism is constantly fulfilling a set of functions or purposes. The fundamental intrinsic function of a living creature is to maintain life through metabolism. But each organism realizes this purpose through its own form of life. To realize its way of life, the organism has to differentiate itself into separate parts that pursue different ends, or organs. Importantly, these parts only exist in relation to the whole, coming into being through the developmental mechanisms organized by the totality of the form of the organism, and existing fundamentally for the whole. Parts only truly exist when serving the whole. A dead part, like a hand removed from the body, can only be equivocally called a hand. Organic bodies are thus bodies consisting of parts that exist only in an intrinsic unity, meaning the parts come into being through and exist for the whole. In contrast, the form of an artifact is generated by the artificer who composes and unites the various materials of the artifact. An axe or house don't emerge out of the natural activities of these artifacts through a developmental process. Nor do they possess their principles of organization internally, nor are they dynamic unities, internally organized and developing themselves. Instead, they are constituted and maintained by people who use their craft knowledge to shape the materials into a form. Their organization is not internal, but exists in the mind of their artificer. Because it lacks a substantial form, an artifact also lacks an intrinsic purpose, an intrinsic teleology. Its purpose is given to it by the artificer. The purpose of tools can even change in different cultural contexts. In contrast, living organisms possess an inherent order and towards purposes, an inherent teleology. Now, there's a further distinction that Marilou Papandreou has helpfully made between things that are artificially caused natural beings and true artifacts. Artifact is a technical term, identifying an object that is brought about by human art that possesses an accidental form, not a true substantial form. However, human activity can also modify substances and even bring them into existence. So medicine is a human art that fosters the healing power of the human body, or IVF leads to a new organism forming, right? So it's not that anything made by kind of techne or technology is automatically an artifact. Artifact is a technical term of a kind of being. Aquinas, for example, indicate that materials like bread result from the activities of bakers manipulating the natural powers of materials, and contemporary Aristotelians point to styrofoam as a new substance. Thus, there's the possibility that a technology can produce a substance rather than an artifact. Okay, so my argument is that these distinctions allow for a better analysis of SCBEMs, but I'm going to approach this argument by first looking at other biotechnologically produced entities, moving slowly from kind of more basic cases to full-on SCBEMs. First, let's take cell cultures, right? In these cell cultures, the individual cell would seem to be what is the fundamental substance. Each cell has its own intrinsic unity among its organelles, nucleus, and other cell parts. It has a set organization, and it has its own teleology, the maintenance of its life through the metabolism of nutrients in its medium. The cell is not an artifact, but a living substance that is the product of the art of biotechnology. The cell culture system as a whole, though, is only an aggregate of such cells. Cells in culture are in communication and coordinate with one another, but they lack a unity, a coherent differentiation in organization, and a shared purpose. If the totality is organized and used in some way, it's going to only be due to the extrinsic goals and organizing capacities of the scientist. Now, to move to organoids, most organoids seem to fall closer to the artifact of cell culture than they do to organisms. Organoids mimic the structure of many organs, but these aren't perfect recreations of in vivo organs. The precise mixture of types of cells generally don't match the natural organ, and the morphology is frequently disturbed. Moreover, directed differentiation often can only lead to the formation of parts of an organ. They are attempts to recreate a part of an organism, a pancreas or a lung, ordered toward a larger whole, the organism. These organs lack the independent functions which are characteristics of organisms. In general, organoids don't even fully perform the function of a part adequately, only recapitulating some aspects of an organ structure. So therefore, they are basic artifacts. Okay, so now to SCBEMs. Now, some of these are clearly parts or artifacts. A lot of the SCBEMs that are out there are closer to organoids than true embryos. For example, gastroloids just recapitulate aspects of somatogenesis of the trunk without supporting surrounding extraembryonic tissues seemed little different from the Keller sandwich experiments long performed by developmental biologists on Xenopus embryos. I think a lot of what we call SEBEMs are hype, right? They're being included under this kind of broader thing that Hannah and Zernicka-Getz are trying to do. The real concern comes with models such as the mini blastoids and perigastroloids that come much closer to showing an integrated developmental process. And these are the kinds of things that we heard last night. At the same time, these resulting embryo models are disorganized. Eventually, their development stops, and differentiation doesn't occur perfectly. The best models also come from the aggregation of different cell types that are kind of differentiated separately and then placed together. It's an aggregative process. So in this sense, they do seem a little bit closer to artifacts than to an embryo or an organism. Yet, these latter SEBEMs do show a considerable process of differentiating into most tissue types. Though they may not be totally successful in development, some show impressive levels of organization with beating hearts, the anterior-posterior axis, most cell types found in the corresponding embryonic stage, and rudimentary organs. Moreover, one might argue that their early failure is due to technical issues and culture conditions that might be overcome through further advances. Thus, one might say that there's a sufficient level of overall integration and a distinctly human developmental trajectory to suggest the form, teleology, and thus unity of a human organism. This argument seems convincing enough to warrant caution about these more advanced kinds of embryo models. While the evidence suggests that most SEBEMs aren't substances, there is a danger that these aggregations may lead to the form and teleology of a unified substance. Therefore, precaution and a respect for human dignity argue for limiting these experiments. The danger isn't that they will become something with moral status as they develop more capabilities, but they're already a human organism. Thank you.
And last but not least, Professor de Graaf, her and her colleagues.
All right. Thanks to the organizers for this amazing conference and for inviting us to speak here. And congratulations to all of you for making it through a host of very interesting and very rich discussions. And it is our honor to be the last speakers of the day. And we'll try our very best to be both engaging and quick. The puzzle we want to put forward, and we in this case is my colleague Anna Pereira-Daoud, Megan Munsey, who is also in the room, and Lien de Proost, who's actually in Belgium and not here with us today, is simple. The field has spent a lot of time asking whether embryo models have moral value, and not enough debating what kind of moral value and what this implies. As you will be aware by now, stem cell-based embryo models are heterogeneous. They have different origins, different levels of completeness, different developmental stages ranging from pre-implantation to post-implantation stages, and finally different purposes ranging from fundamental to more clinical purposes such as, for instance, toxicity screening and disease modeling. And the emergence of these models has led to significant debate about their moral and legal value. There's broad agreement that moral value too is heterogeneous, but there's ongoing disagreement about what features should trigger stricter governance and what cultural limits, if any, should apply. Previously, Lien de Proost and I have argued for a multi-criterial approach to factor in both intrinsic and extrinsic considerations in discussions about embryo models, as we believe this could better account for moral complexity than a unicriterial approach, and it could engage with some interesting arguments that have been put forward in the grey literature on embryo models. However, doing so calls for further conceptual as well as normative analysis, and that's precisely what we do in the chapter that we drafted as well as in this presentation. Now, existing ethical scholarship has mostly focused on intrinsic value, so the value something has for its own sake. But obvious grounds for such value, such as, for instance, consciousness, sentience, are lacking in early stages of embryonic development. So this means that both early embryos as well as embryo models aren't, well, these things don't apply to them. So most of the debate has focused on potentiality. It has featured quite a bit today as well. Yet there's currently no evidence for embryo models that non-human embryo models can actually lead to offspring. And very few argue that complete embryo models possess active potential, let alone incomplete ones. Notably, despite the fact that few grounds currently are available to actually attribute intrinsic value to embryo models, significantly less attention has been paid to extrinsic forms of value. Van Til and colleagues distinguish between 3 dimensions of extrinsic value that we are using to build our analysis upon. The first one is instrumental value. That is, the value something has because it is a means to an intrinsically valuable end. A classic example is money. Then there's relational value, the value something has in view of its relation or connection with other entities. And finally, we can distinguish symbolic value, the value something has in view of what it stands for or represents. A classic example is a flag that might represent our values, our ideals, and our rights in our countries where we live. Now, for many entities, these types of extrinsic value may coexist, conflict, and overlap. Take money. Money has intrinsic value, instrumental value, but clearly it may also have relational value. For instance, when your grandpa saves a stash of money for you over years and hands it down to you as a gift. But it may also have symbolic value because it might represent, again, our national identities and our countries. Now, interestingly, in contrast to in the ethical literature, the grey literature, the governance literature on embryo models has extensively engaged with these kinds of concepts. And we analyzed 3 of them that were published in different countries, namely France, Sweden, and the Netherlands, that built on these extrinsic considerations and found that they draw strikingly different conclusions. In what follows, we'll focus mostly on relational and symbolic value and take a couple of examples in view of time. So, first, differences exist in how the assessment of extrinsic value is envisioned. When it comes to relational value, the Swedish and the French reports, for instance, assume that it's attributed based on the fact that an embryo model or an entity is used in a parental project, and thus attributed by the intended parents, as you can see here. Whereas the Dutch report argues that relational value can also be attributed by society at large. Moreover, reports differ in whether they consider relational and symbolic value a binary or a gradual trait. The Swedish and the French reports assume such values are binary, that an embryo model or an embryo either possesses it or it does not. It is part of a parental project, for instance, or it is not. Whereas the Dutch report contends that it's something that can increase over time, in the case of an embryo, for instance, over the course of a pregnancy. Second, and as a result, differences exist in which entities are thought to possess these different types of extrinsic value. Let's have a look at relational value in particular. The Swedish report distinguishes embryos intended for implantation from those left over from IVF and embryo models, which are used for research only, suggesting that the former have higher relational value than the latter, and also higher protective interests, even if they have the same inherent characteristics. The French report, similarly, argues that embryo models cannot be equivalent to embryos partly because, unlike donated embryos, they were never part of a parental project. The Dutch report reaches a different conclusion. While it notes that little empirical research has been conducted into citizens' views of embryo models, which they take as the basis to ascribe this type of value, it nonetheless concludes that embryos and complete models, which cannot be ruled out from having a similar developmental potential, have the same relational and symbolic value as human embryos. Finally, differences exist in what normative implications are ascribed to extrinsic value. To start, the French report and the Dutch report illustrate this well. Both draw on relational and symbolic considerations, yet, as we will discuss in more detail later, reach opposite conclusions. The French report concludes that precisely because complete embryo models are not equivalent to embryos on relational and symbolic grounds, research on them may be permitted up to 28 days, or the equivalent of it. Well, they propose keeping the 14-day rule in place for human embryo research. The Dutch report, in contrast, holds that embryos and complete embryo models should be subjected to similar culture limits, both 28 days, or its developmental equivalent.
Thank you, Ninka. I will take over from here. So to understand what, if any, normative implications this types of extrinsic value should have, we also looked beyond the governance of research with embryos and embryo models. For each type of extrinsic value, we actually identified analogous cases in which they already play a role in governance. So for example, instrumental value informs laws in which the use of certain tools is either enabled or constrained by the ends they serve, as seen, for example, in organ donation regulations. Relational value informs laws in which entities are for the special consideration based on the relations in which they are embedded. Consider, for example, prohibitions on consuming companion animals. Symbolic value also informs laws, but now in cases of which entities are protected in view of the symbols they represent, for example, prohibitions on the desecration of national flags. These analogous cases are useful precisely because they show us the limitations of using extrinsic value in governance. So for example, instrumental value risks subordinating other values, including intrinsic value, to considerations of utility, as the history of research ethics painfully illustrates. Relational value risks grounding regulatory asymmetries that are difficult to justify coherently, like prohibiting the consumption of companion animals while allowing the intensive farming of other animals with very comparable capacities. Symbolic value is complicated by polysemy, what something symbolizes and to whom may vary across space and time, as debates about flag desecration also illustrate, for example, in the United States. These limitations are not reasons to dismiss extrinsic value altogether. However, they do point to conditions that must be met in order to justifiably ground regulations in such extrinsic value. In our chapter, we identified three. The first one is that these regulations should be able to withstand normative and epistemic scrutiny. The second is that they should be subject to periodic review. And finally, that they should be widely shared and societally intelligible, a condition that is particularly pressing in democratic and pluralistic societies where governance derives its legitimacy from those it governs. These conditions may not be equally easy to satisfy. I see that I'm a little bit over time, apologies. For example, because instrumental and relational value are much more tractable and therefore more identifiable. By contrast, symbolic value might be more difficult to evaluate because it often relies on meanings that are diffused, contested, or culturally valuable. Applying these conditions to the policy reports that Ninka already introduced allows us to assess how well supported they currently are. Such mechanisms and recommendations can fall into two broad categories. On the one hand, procedural, like informed consent requirements, and on the other, substantive, like the 28-day rule that Ninka also previously introduced. Our analysis suggests that whereas the former is broadly supported by these conditions, the latter is not. And to see why not, I think it's worthwhile to look at the justificatory basis provided for such a categorical cutoff point. And I'm looking at Glenn. I will go over this incredibly quickly. So, as mentioned, the French report recommends maintaining the 14-day rule for embryos, but applying a 28-day rule to embryo models with akin development, that possess both embryonic and extra-embryonic tissues. The report justifies this a distinction by appeals to intentionality and origin. Unlike embryos, embryo models are not created within a parental context, and they are also not derived from the fusion of gametes. The problem with these two considerations is that they do not specify why exactly they are normatively significant, and as such, they are difficult to evaluate from a normative and epistemic perspective, and also to share on a societal level.
I think we're going to let that be the last word. A big round of applause, please. All right, now we're going to move to questions. We started a little bit late, so we're going to go till 4:30, and we don't need as much time for the closings, so we'll still get you out on time. I'm a prioritarian, so I want to make sure every person gets at least one question, so I'm going to ask for a question first, Professor Hurlbut. I'm going to give priority to somebody who hasn't asked a question yet. Does anybody have a question for Professor Hurlbut? Nobody has a question for Professor Hurlbut? All right, we'll come back. Maybe people are thinking about their questions. It was too good a presentation. You stunned them, right? We're going to have now a question for Sir Condic. Anyone have a question for Sir Condic? Please. We're going to get you, yeah, from Maureen, we're going to get you the microphone.
There was a mention of the high cost of these IVF procedures, and it troubled me, partly because I did some of the original research in the 90s showing that in Massachusetts, the mandate raise cost to $1.71 a month. And currently, it sounds like it's about $3 to $5. So I get very troubled when I hear this sort of IVF is expensive when we look at it individually instead of across the way. We don't hear this about knee replacements, hip replacements, which per procedure cost more. So I was just wondering if you wanted to comment on sort of more, as opposed to the per person per cost, the societal cost were this to be covered the way many other medical procedures are.
Right, I think I was not given an opportunity to comment on knee replacement, and I probably would have the same objection to that procedure. But I think it is relevant when you consider not the across society cost, but about the access question. So the fact that the procedures are expensive impacts who is it that can in fact access them if they do not have insurance that covers this kind of very expensive procedure. So if I'm uninsured with respect to IVF and I don't have $15,000 to $20,000 a pop, I can't do it.
My apologies. I wanted to be clear that this is partially a problem because we don't have the insurance coverage that we have for so many other medical procedures.
Right.
And I thought that point needed to be made here and in other panels as well.
The observation is simply that this question of equity has been raised about IVF relative to the cost of more fundamental kinds of healthcare.
Great. We've got a question for Professor Suter and Professor Cahn. Any questions? Please go ahead. Put you the microphone. Give me 1 sec. Go right ahead.
I have what may be a more technical question. You noted that for asexual reproduction, the resulting human embryo or child, depending on the scenario, would result in 75% genetic likeness to the progenitor. And I was wondering if you could dive into some of the science behind it, because I've heard it framed as cloning typically. And so, yeah, I'd love to hear your thoughts.
I'm so glad you asked that question. So the difference between cloning would be the exact same. So let's say you have A, B for every gene, right? An A version, a B version. If you cloned yourself, it would be an A version, B version for every single gene. It would be the same genetics. But when you do IVG, each time you're flipping a coin, are you passing, is the egg going to be an A and the sperm going to be a B, or is it going to be AA, BB? And so it's not always going to be exactly 75%, but sort of if you were flipping a coin, it would be like that. Yeah. So that's the reason it's not exactly a clone. And I think that's really important, but there is that consanguinity concern because there's a much higher risk of recessive diseases actually manifesting because you'd have two copies.
Sure, very quickly, yep.
Under state law governing human cloning, would that 75% potentiality still qualify based on what you know of the law, or would that be considered distinct?
I mean, I think you'd have to look at the laws, but I think it would be distinct. I think the way that cloning is defined would not include this. So I really think it has to be distinguished from cloning. And I was almost going to say that sentence, but I was trying to get under 10 minutes, so.
But I'll just say the flip side is that the existing appropriations right, or the Adderholt Amendment, does probably, according to FDA, cover IVG, but may not cover cloning. Right? This is the view of Peter Marks when he was at FDA, that it was covered. But
he said it might be.
Right? Well, I think,
he said it might be. He thought IVG might take the spirit of it, but I think a technical reading of it wouldn't. But
there's an open question. We love that. All right. A question for Professor Schurz. Do we have a question about artifacts? I'll ask a question, which is I was very interested in the question about intention's role in the analysis, and this has come up a few times. But at times I thought your view would be that the intention, I thought about the analogies to the function and making a function that somebody has an intended function for something, whether intention plays a role in the categorization that you presented?
No, I mean, for artifact, the intention of the artificer plays a role in what the artifact is used for. But once something becomes a substance, it has an internal teleology, that the purpose is intrinsic to it. So at that point, the intention of the maker has no further role.
Great. Did you want to follow up on that? We'll get you.
Yeah. So, and I'm going to read your paper again, so this is probably not quite right. But a substance that is built by an artificer, okay, would it inherently always be artifice, or?
No, because you can have, an artificer can modify something, a natural being. Like, so a doctor will use the art of medicine to kind of bring you back to health, but you will continue to be a substance in your own right. But then artifact is a more technical term of something that isn't a substance itself, that's aggregated, put together, and forced together by someone who makes it.
Okay, thank you.
Great. A question for Professor de Graaf and colleagues. Do we have a question? Go ahead.
I'd love to know what the other reports said about the informed consent. How did they how did they support the informed consent element? You were contrasting that with the 28-day categorization, or categorical exclusion.
So indeed, the informed consent aspect, they often recommend, specifically for more complete models, to actually make it more strict. So actually people have to be informed that their cells would be used to create these models, for example. This is also something that the ISSCR recommends, and that is being enacted in the Netherlands, for example, already in, pending legislation. Does this answer your question? Thank you.
Maybe if I could add one point, and I see Megan also wanted to comment, but what was also quite striking to us is that when you looked at the way in which extrinsic value was being attributed in these reports, actually none of them considered the possibility that there might also be relational value that a donor holds. So a donor can also attribute relational value to its cells or the embryo models created with its cells. I think that's actually quite widely recognized and also one of the bases for informed consent, but it wasn't actually mentioned as a form of relational value.
Megan, did you want to jump in? Great. Take your question 1 second. I'm just going to, now it's free range. Anybody can have a question. Go ahead first. Please, go ahead.
So I love the sort of the comparison of the reports, and I was curious in the differences of the reports. Is it a normative difference, or is it is it unique to this area, or is it just that in some governance models, like in France, is more instrumental, and there's a more relational approach in other countries?
I would say it's most definitely a normative difference. There's also conceptual differences. I think you could argue that some of the differences that we highlighted are conceptual, but definitely major normative implications, right? Because they actually end up justifying different culture limits, for instance, amongst others, based on these kinds of considerations.
I want to follow up on this, which is to say there's a way when I read your paper that I thought a little bit about Julius Caesar, where I come to bury Caesar, not to praise him. And there's a way in which it seemed as though the more you started unpacking the different kinds of extrinsic reasons, the less persuasive I found them to be as reasons in relationship to intrinsic. I was curious whether that was a hidden ambition of the paper, a feature of the paper, or whether that was an accidental byproduct of me, the reader, imposing that on that.
So you found them less and less convincing as
the more
arguments?
you entangled them.
Yeah. I would more or less agree with that, although I do think that there's implications that hold up. But yes, so the original paper that Lean and I wrote was a short commentary, and there we just said, well, we need to think about this. And then it's been taken up by some people to say, well, all of these considerations are therefore relevant and must therefore be honored. And that's not definitely not what we're arguing. We're just saying we need to talk about this, and we need to scrutinize them, and that's what we've tried to start with in this chapter.
Great. I think Professor Kresher, you had the next question. Go ahead.
Yeah. My question is for Sonia and Naomi. You guys do a lot in what will be 5,000 words. But I wanted to ask you about the map you put up and and how you are thinking about it. So let's say that we end up with the patchwork that you say might map onto what we see for gender-affirming care or abortion. Unlike those types of health care, maybe, right? If I were thinking about IVG, I think of a future where you could do a cheek swab, put it in an envelope, send it to an IVF lab in another state. They create the embryo, they do the fancy whatever they do to make sure it's safe and shipped right back in to the state where I live, and then a clinic transfers that embryo into my uterus, right? So I guess I'm sort of thinking about access and barriers. To my mind, they would be they would cash out very differently with IVG than when we think about abortion and gender-affirming care. But I just wondered how or if you have thought in any more detail or carefully about really how that ban on IVG would operate.
Yeah, no, that's a really good question. I guess I wasn't quite imagining it the way you were describing it, although it depends where we're talking about in the future, right? Because at the moment, I'm kind of imagining you have to go, somebody's taking the cell from you, that it wouldn't be as easy as a cheek swab, and that you'd probably have to do a biopsy, and so it would be more of a medical procedure, and so you would be in the context of a clinic. And so I think in that context, it would be easier to sort of control in the same way that we think about abortion care pre-mifepristone. But yeah, I guess in the future, I'm always skeptical about how much you can just like take a cell from a dead person or how much you could just do a cheek swab and whether that would work. I just don't know the science well enough. But then I think it would look different if we were talking about that kind of world. That's a really interesting question.
Jinping, I think you had a question. Go ahead.
I feel like my mind went together with Katie's. So my question is also for Sonia and Naomi. I was thinking, the map for IVG, how similar it is to, the map of abortion and gender-affirming care. Probably you remember that Glenn's 2022 or 2023 paper said that those who oppose abortion may not oppose embryo destruction, vice versa. So do you think it might be possible that states like Colorado or California, they really want to protect abortion, but because they are highly democratic, so they feel that it's safe in their state to regulate IVG. So the map might differ on those issues. Yeah, thank you.
So if I'm understanding your question, you're asking if states that are sort of more pluralistic would regulate, or I actually imagine a state like California, and maybe if it's proven to be safe, right? I'm assuming safety here, that it would actually encourage or incentivize it. So I saw it as sort of some of the blue states that might try to make it more easily accessible. But yeah, it doesn't necessarily map perfectly onto abortion, but I do think on the gender-affirming care, I think a lot of what we're thinking about on restrictions could overlap, and I do see some parallels with abortion as well in terms of the sort of different issues with abortion. The gender-affirming care, I think, sort of the challenging the a-traditional kinds of families and just ways of being.
I wanted to comment on Sonia's answer because I think it alluded to something that none of our discussions have touched on, which is the safety issue. Given where we are with IVG, even if we could translate this technology from mice to humans directly, the only way to confirm the functionality of those in vitro generated gametes would be to make babies out of them. And not just make embryos, but implant them, have them come to live birth, and ideally let them mature to sexual maturity, because that's the only way we know that we've gotten normal individuals. And when you look at the statistics from mice, not only was it very, very hard to get oocytes out of this procedure, but even when they got oocytes, only a very small number of them fertilized and produced embryos. And when they transferred those embryos, only 1% made it up to live birth, which means 99% of the 1% of the 1% died or were extremely abnormal. So are we going to do this with humans? Really?
I'll be even more provocative to say, what if we'd asked the same question before IVF? Would we have had a similar conversation or a very different conversation and how to think about it? But did you want to jump in on that?
Yeah, because I also had that same question for Sonia, although Maureen put it as a statement. What does safety mean? And I mean, basically we don't do clinical trials that subject people to potential harm unless there's a good reason. And this is a situation in which you can only get harm, you in effect can't get benefit because you don't have an existing person who's in need of some kind of care and therefore it's worth taking some sort of risk or whatever. You basically have to make children who are experimental in order to even ask the question in the most conventional way we ask questions about the safety of biomedical. So my question is sort of what, with that as a sort of background to the hypothetical scenarios, but let me just ask one further question, because it seems to me that unless one leapfrogs over this safety issue, IVG isn't for reproduction, it's for something else. And yet it's being developed as though a reproductive technology. But, you know, I mean, the bottleneck for many, many things has been access to eggs. And
I'm going to pause you, Sonia, on the answer. We're going to get Megan's question, then we're going to finish. So we'll get answers on that, both of those. Go ahead,
I think
Megan.
I had a question like about 5 comments ago, so, but I'm going to just maybe just use this last comment to say that we've talked a little bit about MRT, and I think I agree with you. IVG is so far away. It's good to think about it. It's good to, and to your point, Katie, you're right on the money. You know, it's a vial of cells. It's a bit of blood. All you need is a little bit of blood to make stem cells. The big question is, can you then make gametes? But leaving that aside, I just wanted to make a comment to say, yeah, the MRT debate in Australia, we've had our law change recently. There's a big experiment going on. So, you know, watch that space. They've introduced a national framing body that's overseeing an experiment on children where we'll be watching how it unfolds over the next 10 years. So it's a government-funded clinical trial. So that's one model. It's very transparent. There's a lot of thought going into it, but you might be interested in that.
Great. Sonia, you get the last word.
Well, I was going to say what you said in the first response to the question, would we even have IVF if we had raised those questions, right? So, but also, I guess I have a question about your question. I think IVG is about reproduction. It can be about other things, but it does allow for certain kinds of reproduction. So same-sex reproduction is not possible genetically without IVG, right? So I don't think it's just, but it can obviously be about other things, or for people who can't produce gametes, it can be about reproduction. But yeah, the safety question I think is a huge one. You know, I'm very ambivalent about IVG precisely for that reason. Precisely for that question, because I don't know how we can determine it in a safe way. You know, trying to reset the epigenetics and have that work, and I think there's some really, really big questions. But there are also a lot of really interesting questions if we assumed away the safety problem, which I realize is a really big thing to do, assume away something very major like that.
Please join me in thanking this panel. And now I'm going to call up my co-editors, and we're going to say a very, very brief closing.
Oh, yes, such a pleasure to meet you. Too. Thanks for bringing in all these different perspectives.
All right.
Hello. I was waving at you all across the room today.
So you heard from us at the beginning, and you get to hear from us very briefly at the end. I'll go first and just say a huge thank you. This was exactly the kind of engagement we wanted. The papers are fascinating. They're going to be even more fascinating when they're 5,000 words, including footnote. Just saying, they're going to be more fascinating once we've had a chance to reflect on what's been said today. I'll turn it over to my co-panelists in case they want to say something else.
Yeah, no, just very briefly, I wanted to join Glenn in thanking, first of all, my co-editors and the team here, but also you all. It was such a wonderful and rich conversation, and it, I think, achieved what Glenn's vision was, namely to have people, different disciplines, different backgrounds, different perspectives, most importantly, engaging in a fruitful and charitable way, and I think it's cause for optimism for the future for me.
Thank you so much. Coming into this, I didn't think it would be possible for me to say that this exceeded my expectations in every way. And just trying to think about some of the themes that emerged from today. One for me is everything old is new again. I was surprised at how many times the Davis v. Davis case was invoked. Now it's in its 36th year, and yet we seem to be coming back to it in meaningful ways. And also, Dove isn't here, but the things that are new are better interpreted through things that were old. And again, I see that theme having been processed over and over again. I mean, certainly what we learned today is that scientific advances surrounding reproductive ingredients bring together a lot of different concepts and disciplines. I mean, just naming a few, certainly we talked about definitions, about status, about law, about philosophy, theology, sociology, business and industry, even aerospace. Who knew that that would come into play? But it just really, I think that the message I'm going to take away is that we should do this again, whether we do it soon or well into the future and our successors thereafter. Certainly such a courteous and value-laden experience is something worth repeating. Thanks so much for letting me be a part of it.
I echo all of the thrilled feelings about the day. I want to say we will follow up with some additional information for all of you as writers. Give us a couple days. We've been doing a lot of stuff. We're going to eat ice cream and sleep, I think, until next week. But we can't wait to stay in touch and to see how this project develops. I have been thinking about my father today, who is an English professor and very much the grammar police in our house, because what I keep thinking is how very unique today was, which he would have said is not a thing. You're either unique or you're not unique. You can't be very unique. But I think we managed to be very unique. There's a sense in this room, I think, of wanting to continue the conversation at a time when a lot of people are avoiding, perhaps, the conversation. And I think I want to just conclude by, maybe one other language grammar question, which is, I was struck by, at core, how many people, whether explicitly or not, were wrestling with this question of what the embryo is. And, to quote, well, it depends what the meaning of the word is, I suppose. I don't know if that's an appropriate reference here, but it does. It's really a question of the different perspectives in the room and how we're all wrestling with a lot of the same questions, even if from very different places. Thanks.
Safe travels, everyone. We'll see you soon. Thank you for our friends on Zoom.